First talk, we will have Dr. Larry Hamann, who is the co-founder, president, and CEO of Interdict Bio, where he pioneers small molecule context-dependent translation inhibitor to tackle historically undruggable proteins, particularly in cancer and neurodegeneration. Larry earned his PhD in organic chemistry from University of Michigan, and he has an impressive over 30 years of experience in drug discovery, where he has led teams to successfully advance more than 22 molecules into the clinic, including approved drugs treating diabetes and HCV, and also has worked on targeted protein degraders that are in phase 3 clinical trials currently. He career also includes significant roles at Celgene, Novartis, BMS, also Takeda, and he is a prolific inventor, author, and a trusted advisor for many biotechs and VC firms. His contribution to the field have been recognized with prestigious awards, including ACS Heroes of Chemistry Award, the ACS Division of Medicinal Chemistry Award, and also he has been inducted into the Medicinal Chemistry Hall of Fame. So thank you, Larry, for agreeing to present today, and the floor is yours.
Thank you for the very kind introduction, Hubert, and it's a real privilege to participate in this series, which I've been a big fan of since the very beginning. As Hubert said, was a slight departure from the typical topics for this, but I think you'll see the similarities, and I'm excited to share what we're doing. So let's see here. If I can. Okay. Great. Thank you. Let's see if I can just, there we go. Yeah, it's a pleasure today to tell you about what we've been doing at Interdict for the last just over 3 years. Um, as mentioned, if you look back across the last couple centuries of small molecule drug discovery, really for the most of that period, it was really all about natural products. And it was only in the 1970s when some of the very first designed inhibitors came onto the scene, notably captopril, an inhibitor of angiotensin converting enzyme for treatment of hypertension, and shortly after that, Gleevec as the first kinase inhibitor for CML. And then, really, for several decades, small molecule drug discovery was mostly about inhibition of targets that had catalytic function that could be abrogated. And then along come degradation therapeutics, which this audience is very, very familiar with, where a single molecule of drug can address hundreds, if not thousands, of molecules of the target of interest, given the catalytic nature of the way that they work. And of course, the first and somewhat notorious entrant in this category is thalidomide, which nonetheless was repurposed as a treatment for multiple myeloma and led to follow-on molecules like lenalidomide, pomalidomide, and now quite a number of additional molecules advancing through clinical trials, as well as launching more than 20 biotechs. And of course, heterobifunctional degraders following on from that as well. And we imagine what could be more powerful than getting rid of a disease-causing protein after it's formed, but to prevent it from being formed in the first place. And so we're imagining that the next quantum leap in innovation in this regard is interdictors. And the name comes from the military usage of the term interdict, meaning basically to destroy enemy resources before they even get to the battlefield. So just to reorient you on the mechanics of protein translation as I begin here, here I'm showing the two subunits of the ribosome translating along messenger RNA, and these transfer RNAs are bringing in each successive new amino acid to add to the growing chain until the stop codon is encountered, release factors are recruited, and the peptide chain can be released into solvent and folded into its proper shape to perform its function. And because the ribosome itself is energetically costly for the cell to manufacture, it gets recycled and reassembled on another molecule of mRNA to continue translation. Well, now here I'm showing translation occurring as before, but now we're going to see an interdictor molecule bind in a pocket in the ribosome and exert selective friction against the sequence of a peptide chain as it's moving through, causing arrest of translation, ribosome collisions, and ultimately the cell degrades the mRNA and gives up on the process, and so the protein never gets formed. Well, there's actually precedent for this modality in an approved drug that's called Cinrybo, that is a natural product homoharringtonine. This was discovered and developed by Cephalon more than 10 years ago, and it's used in second line for CML after failure on Gleevec, et cetera. And the molecule is not a particularly selective molecule. It's rather large. It occludes the entirety of the peptidyltransferase center of the ribosome, and as such, it's not particularly selective. But nonetheless, it had adequate safety and efficacy to be an approved drug. But a more meaningful precedent came in this seminal paper from my two co-founders at UCSF, Jamie Fraser and Denisa Fujimori, in early 2022, where they showed in bacterial ribosomes this historical Pfizer antibiotic molecule linezolid, which had historically been thought to be a global inhibitor of translation in the prokaryotic ribosome, was actually a sequence-selective inhibitor. And by using biochemistry and cryo-electron microscopy, they could show even, for example, that mutation of this alanine to a glycine ablated all of the antibiotic activity of this drug. And so this was a precedent for us to imagine pursuing selective inhibition of translation in eukaryotic systems for therapeutic benefit. The interdictors draw upon many of the virtuous features of the most advanced small molecule therapeutics. These all happen to be molecules that I had the pleasure to be part of the discovery and development on in prior teams and other companies. But like RNA splicing modulators like branaplam, these our interdictors are intervening at a point in the target life cycle where there's a very favorable stoichiometry. The copy number of mRNA that's being translated is a lot lower than the copy number of the eventual target protein. Like molecular glue degraders, we're hijacking native cellular machinery to perform functions for therapeutic benefit, but instead of using the ubiquitin proteasome system, we're intervening at the ribosome. And again, very small drug-like molecules that are very amenable to optimization for oral delivery. And then lastly, like heterobifunctional PROTAC type molecules, we are heterobifunctional as well, in that one side of the molecule is binding to the interior of the ribosome, which is comprised of ribosomal RNA. The other part of our molecule is binding to a sequence of amino acids of that peptide chain as it's going by. But unlike PROTACs, this proximity is guaranteed. The peptide chain has to go through the peptidyltransferase center in the ribosome. We don't need a linker of any sort to bring these together. We're just creating the stickiness to prevent movement. So, if I drill down further into, translation inhibition, here is just a schematic showing normal translation where you have ribosomes spaced along a messenger RNA, and this translation proceeds as expected. Cinrybo is this homoharringtonine molecule has because it occludes the entirety of the tunnel, it stops every translating ribosome at the very first residue, and so this is an initiation defect in a sense. I'm sure all of those on the call today are familiar with GSPT1 degraders. Well, GSPT1 is also known as ERF3A, the release factor to release translating the peptide into solvent from the translating ribosomes. And degradation of GSPT1 essentially causes a pileup of all ribosomes at the stop codon. And so again, not selective at all. So in our sense, we're intervening in the elongation phase and relying on intervening at certain sequences to selectively stop translation of targets of interest. So we didn't have to go to any kind of high throughput screening exercise to find starting points for chemical optimization simply because there are so many known molecules that have been studied by the academic community over the past several decades, most of them natural products, which were retrospectively characterized because of an interesting phenotype. Notable among those are, as I mentioned, Homo harringtonine, and all of these have been characterized through either X-ray or cryo-EM as binding in this similar pocket in the ribosome. The one I want to focus on today is a nisomycin, a very ligand efficient and compact molecule that is binds in this part of the A site of the peptidyltransferase center to block translation. And this was a nice chemical starting point for us to explore elaboration to induce, better selectivity. So the lowest hanging fruit that we pursued as we were starting out were to make simple derivatives of anisomycin that swap this ester moiety for a carbamate for greater plasma and solution stability. And by appending moieties that were projecting towards this peptide chain off of the anisomycin core, we could design those that had a basic moiety that would be predicted to have greater engagement of acidic side chains such as glutamic and aspartic acid, and or nonpolar groups projecting towards that peptide chain that would have better affinity for things like alanine, proline, lipophilic residues. And here I'm showing the results of some ribosome profiling data sets where we basically stop every ribosome in its tracks, and the part that's protected inside the ribosome tells you what sequence the molecule was stalled on. And as predicted by our computational and structure-based design, the molecule with the basic group has a very strong preference for glutamic and aspartic acid sequence, and the nonpolar group for alanine and proline. And these preferences are increased as the concentration of the molecule declines. So furthermore, we could take artificial constructs of a 6 times former of the preferred sequence of a given molecule and show that we could achieve even 100-fold selectivity for a given sequence in a length-matched translation assay. So because of the richness and diversity of the side chain composition of the various amino acids, we can find great starting points for intervening for sequences of interest. And if we look at this 2D map of sequences for the first 50 or 60 amino acids in the N-terminus of targets of interest to us, like MYC and MCL1, you can see just visually they're quite different from one another based on these characterizations. There's furthermore, of course, a steric component to these amino acid side chains as they're gliding over the molecule that's anchored in the ribosome that is part of the friction that is applied to that peptide chain in a selective way. And then, of course, you can imagine some of the very extreme sequences that characterize things such as mutant Huntington protein and other poly-Q expansions in the N-terminus. And then, of course, C9orf72, which is implicated in ALS, that has this duplex repeat sequence in the N-terminus of proline arginine. These represent very unique and addressable sequences by our modality. So we also have heavy reliance on cryo-electron microscopy. We've already solved more than 25 in-house structures to date. We collect the data sets at the University of Michigan's facility. Here I'm going to show you a couple of structures that are at lower than 2 angstrom resolution. And as we zoom in on the ribosome, you're going to see a couple of things. First of all, in blue is the ribosomal RNA. In green is the transfer RNA that is bringing in the next amino acid. And then the salmon color is a sequence of the oncogenic transcription factor MYC that we have, that is this preferred stall site for our molecule. And this is arrested in mid-translation. And this is EQEDE. So again, very rich in glutamic and aspartic acids. And as we continue here, you're going to see this is the conformation of the peptide chain in the absence of an interdictor. Now, with our preferred interdictor here, you can see the reorientation of the side chain of the aspartic acid, as well as other nucleotides in the pocket that make accommodations based on predicted interactions from our small molecule. So, from these two cryo-EM structures, we can gain a lot of valuable information that guide our future structure-based design efforts. So a lot of the inputs that we generate through either cryo-EM or the ribosome profiling, et cetera, can be fed into a machine learning algorithm that we've created called the interdictionary that help us identify appropriate places for intervention with our molecules. We can do a very rapid analysis of across the entire proteome to find the best opportunities for applying this technology. But from a therapeutic standpoint, we have two kind of orthogonal use cases that we have been pursuing since we started the company. I'll mostly be able to talk about one today because of time constraints, but basically, the first is these very short half-life oncogenic drivers. And so, one of the hallmarks of proteins like MYC and others is that they have extremely short half-lives relative to the rest of the proteome. Here on this logarithmic scale on the bottom, you can see MYC as an extreme outlier relative to the typical median half-life of more than 40 hours in HeLa cells and 90 hours in B cells. The other use case, as I kind of alluded to before, are these long repeat-driven proteinopathies that are characterized by either polyQ expansions or et cetera, and that are part of neurotoxic proteins, that if we could block the translation of, those proteins never get formed and can never become part of an aggregate. So if we think about MYC as the target, which is our lead program, MYC is of course needs perhaps no introduction to this audience as a master regulator in so many different cancers. It's really a driver in about 90% of all cancers, including those with MYC amplification, which account for about 20 to 30%. There's really no direct MYC targeting agents in the clinic, and there could be problems anticipated with all of those because of its rapid turnover kinetics. But of course, it's still a very important target to go after. It's, as I mentioned, there's no direct targeting agents, partly because it's intrinsically disordered, as you know, and here's an AlphaFold predicted structure, but there are really no available binding pockets that could be addressed with either small molecules or biologics. But I would argue that the bigger problem with MYC is actually its turnover kinetics. So MYC has about a 15 to 30 minute half-life in most cancer cells. So any kind of an approach that would rely on occupancy would be stoichiometrically overwhelmed by a constant influx of newly synthesized MYC, making it impossible to keep up. And this has been, in part, the reason that approaches in the past have failed. But this actually becomes a liability for MYC when it's being addressed by a molecule of our interdictor modality. And here I'll illustrate that in this following metaphor. So if we think about protein homeostasis as the emptying and the filling of a bucket, again, interdictors don't do anything to deplete the existing pool of a target in the way that a degrader would. They merely turn off the tap to new synthesis. And because very, very short half-life proteins drain very, very fast from that bucket, when we turn off the tap, those proteins are gone within minutes to hours, and addicted cancer cells cannot survive for more than a few hours without their preferred target before they get cast irreversibly down the path to apoptosis, et cetera. And if we happen to have a partial slowing of the rate of translation of a couple of other targets during that time window, we expect to have an insignificant phenotype because of the very slow rate of draining of typical targets in the proteome. And I'll illustrate that here. I'll walk you through this experiment we did to look at washouts that better simulate the exposure profile of a molecule in an animal or in a human patient. One of the problems with cancer cell viability assays in vitro is that they're a closed system with a fixed concentration of molecule in for the whole 72 or however long, how many hours the assay goes on. We wanted to simulate a pulsatile interruption of translation. And so here, what I'm showing is two different cell lines with our molecule, an MCF7 MYC-dependent breast cancer cell and HEC293 normal cells. And when we treat with drug for 6 hours, wash out for 18, and then repeat this on a daily basis, what we can see is we have a persistent growth defect in the MYC-addicted cells where any partial slowing of the rate of growth during that first 6 hours that is experienced in the HEC293s immediately returns to exponential growth upon washout of the compound. So this temporal aspect of how we can interrupt translation in a pulsatile way is a big factor that is even more important in oncology settings for these short half-life targets than the actual sequence selectivity that we obtain. So we've developed a hybrid assay that can look at translation rates. Here, just showing an example for how we can measure again, the rate of translation of a given construct. We can look at the loss of the target of interest by Western. Here I'm showing in a 22RV1 MYC-dependent prostate cancer cell line that we can rapidly deplete MYC in a timescale consistent with what we are aware of MYC's kinetics in this cell line. And it's also gratifying to see that a MYC-regulated gene, cyclin D, is also depleted in this time window, corroborating the mechanism of action. And of course, this translates into a great cell killing in a typical cell viability assay. Because so many it's hard to find a cancer cell line that's not MYC dependent. Here I'm showing a panel of cancer cell lines that we have studied, and those with no coloring are just, it's not in the depth map, but of course, it doesn't mean that they're not MYC dependent. So we have such a great, 10 nanomolar to 100 nanomolar EC50 on all these different cancer cell lines, and we don't really touch PBMCs at all. So I talked about 22RV1 prostate cancer cells as one of the more important ones that we can address, but also for the next couple slides, want to share some data on this MDA-MB231 cell line, which is a triple negative breast cancer cell line. So we optimized some of our molecules into early tool molecules that were suitable for in vivo study. And in this xenograft, in this triple negative breast cancer line, we're showing at a low oral dose, we can get exceptional tumor growth inhibition. This is a very difficult to treat cell line. Typically, the only options clinically are radiation, chemotherapy, surgery. Keytruda is now being approved for triple negative, but again, it's a very difficult cell line. When we take tumor samples from these animals at the end of the study, we can see that by RNA-seq that the MYC-regulated gene sets, and there's about 60 in this top set here, are the most downregulated genes, again, corroborating our mechanism. Just for comparison's sake, again, the only thing that really works in this cell line preclinically is paclitaxel. These other agents don't have any appreciable activity. And a more optimized molecule that is moving forward. And again, I particularly chose this study where we allowed the tumors to get to a significant size, close to 500 cubic millimeters, before we increased the dose to show we could induce shrinking of the tumors and regression. Very, very well tolerated from a body weight standpoint. And what we might expect as a sentinel toxicity signal in such an assay would be related to what has been seen again with GSPT1 degraders, where rapidly proliferating cells like myeloid progenitors should be affected first. And here we did a complete blood count panel at the end of this 40-day tumor study and show zero evidence of any cytopenias whatsoever. Furthermore, again, there's in addition to just the wide range of solid tumors and heme-onc settings where this can be applicable in the clinic, it's particularly noteworthy that we can address some key resistance situations that have occurred. If you think about the CDK4 inhibitors like palbociclib, one of the main resistance mechanisms is upregulation of MYC. And of course, about 70% of patients treated with palbociclib ultimately have progressive disease by 40 months. Here we can show that one of our interdictor molecules is equipotent on the palbociclib sensitive and palbociclib resistant cell lines. And again, this bodes well for additional clinical applications. In the interest of time, I don't think I'll be able to share with you about our efforts in neurodegeneration, but suffice it to say, we have molecules that are able to stall the polyglutamine repeat expansions in mutant Huntington disease. Here I'm showing a cryo-electron microscopy structure that shows a molecule that was designed to specifically favor interactions with glutamine is able to do that. So anyway, again, thank you for the opportunity. It's a pleasure to participate in this session, and I just want to thank my amazing team who has been just tireless in advancing this cutting-edge modality. I also want to thank my co-founders, Dennis Fujimori and Jamie Fraser, who have been wonderful to work with, and our scientific advisors and collaborators. So thank you.